All right. Well, welcome everybody. This is the first webinar of the year that Innovate Bio is sponsoring and putting on. We are grateful today to have Bijoy Desai talk to us from the Align Foundation. He shared some of the information about the tournament with us this summer and the Antibody Engineering Hackathon, which we are thinking about doing another one in August. So be looking for information about that. And this is a really exciting opportunity, we think, for you and your students. And there are even some funding opportunities, which I'm going to put in the chat again here. There is a presidential AI challenge that will provide some money to teams of students that are working on learning about AI or using AI. And you can look at the website and learn about that. There is also the National Science Foundation also has a sorry, I put these in both. They're kind of on top of each other. I'll put them in again. The National Science Foundation also has a supplemental funding opportunity. It's called a Dear Colleague letter, and the Dear Colleague letter provides up to 20%. You can ask for up to 20% of your current grant funding if you have either an ATE project grant or you have an excellent grant. So those are some great opportunities, I think, for incorporating AI. But the second one has to be with high school students. They want to see collaborations between community colleges, high schools, and industry, and you have to have high school students, just to make that clear. All right, so let's hear about this great opportunity from the Align Foundation. We will have all the information on our website. And Bijoy, I'm going to let you take it away.
Awesome. Thanks, Sandra, for the introduction. And I'm very excited to talk about our upcoming protein engineering tournament. Let me just share my screen here. Can everyone see? Great. Awesome. So a little bit of introduction about myself. I'm Bijoy Desai. I'm a technical program manager at the Align Foundation, and which I'm going to talk a little bit more about. And I'll be talking mostly about our community benchmarking initiative, which is one of our community benchmarking initiatives, the protein engineering tournament. So we have organized and we are ready for the upcoming protein engineering tournament, which is a global community-driven benchmarking initiative for predictive and generative AI models in protein engineering and design. Our registration opened in July, and we already have a large number of teams that have registered, and we are still accepting registrations. So we have about 235 teams so far. It's a global initiative, so we have a total of 30 countries around the globe. Every continent. We just don't have a team from Australia, but otherwise, every continent is represented. We have academic teams from Nobel Prize winning labs to community colleges, as well as industry teams ranging from startups to established big pharmas and biotech companies. Yeah, so very excited to announce that. And I'm gonna spend the next 25 minutes just going telling more the structure of the tournament more in detail. But before I do that, I wanted to give you some introduction of who we are, the Align Foundation, who we are, and what kind of things we do. So the Align Foundation is a new kind of science funder, and we recognize that the main bottlenecks in the age of AI in integrating AI in biological models biological engineering or other biological models is the access to good quality data, as well as automated research methods, methods that are compatible with generation of those kind of good quality data. So we were founded in 2021. We are a global team, and then we have even larger diversity of collaborators. So we have 150 external collaborators, and we have we routinely put out requests for proposals, and then we publish. We are open data kind of organization, so we routinely publish our results. We also have our data portal on our website where you can access different kind of data sets that we are onboarding and generating. So that's a little bit of a line, just give you some idea of like what kind of activities we embark on. So the main thing that the three initiatives that we do is road mapping, data acquisition, and benchmarking. Um, in the road mapping, we basically try to align the community on what is some of the top challenges in terms of generation of large data sets. What are those problems? And hope that experts, subject matter experts, researchers in both academia and industry can come together and identify what is some of the top challenges in data generation. And we do that by putting out RFPs and holding workshops, road mapping workshops. Um, and then once we have aligned, we basically chart out a project plan and uh, our open data sets initiative basically acquires large data sets that are open source, high quality, reproducible, as well as open source. Um, and we do that by having our automation partners, as well as having sample logistics. And then finally, we test the utility of these data sets in training biological AI models, both predictive and generative, by holding transparent benchmarks. So we all know the value of CASP, what it had on improving predictive models in structure prediction models. Um, and we hope to do the same for various, like both protein function as well as other predictive as well as generative models. So here we basically want the community to come together and we set out a certain challenge and uh, with specific tasks and specific metrics that the community comes together and we compare AI models side by side and see how, well, not only where the models currently are at in terms of their strengths and weaknesses, but also what is the gap in data. So sometimes we also know that the data itself is limited. So that informs further road mapping and data generation. So it's almost like a virtuous loop of identifying or seeing where the AI models are at, and then identifying the gaps in data sets and acquiring new kinds of data. Um, so, and then that kind of is a good segue to the community benchmarking initiative. One of the community benchmarking initiative is the Protein Engineering Tournament, which is benchmarks predictive and generative models in protein engineering. Um, so basically, our ideal goal is to get to a state where we don't need experiments and we can just use predictive and generative models to solve some of the biological engineering problems, particularly in protein and enzyme engineering. Um, but before we can get there, we need to really understand and assess where do the predictive and generative models in AI are at and what are the gaps that we need to fill. So that's where these community benchmarking initiatives uh, are are the most powerful at. So one of the most novel thing we do, unique to other similar initiatives, is that we provide novel data sets that are high quality, uh, that can be used optionally by the participants for training their models. And so they can use that data set. And these data sets have multiple parameters. So oftentimes real world protein engineering exercises, they require multi-parametric optimization along activity, thermostability, and many other parameters, as you might imagine, for its use in real world. Um, so in the predictive phase, again, we provide the optional data set that the participants can use, but then they're also free to use their own data or publicly available data to train their models. And then we compare them side by side by having open and transparent metrics, evaluation metrics by which they would be assessed by, and that relate to real world protein engineering tasks. And then for the predictive round, they predict the properties of a withheld data set where we only provide the sequences. We have experimentally determined the different properties, and then the participants have to accurately predict those properties. Um, and then that's what we use to generate the leaderboards. And then the way we have organized it is organized as a tournament. So the top participants advance to the next phase, which is generative phase, where again, they can use the optional training data, but they're generating sequences with enhanced properties, not just along a single parameter, but multiple parameters. Just like I said, like for example, temperature, thermostability, activity, etc. And then what is unique here is like we take all of those sequences and we ourselves experimentally validate and assess side by side the participant submitted sequences. And then that's what we use to generate the leaderboards and award the winners of the tournament. So just to give you an idea, we have had a pilot tournament that was held in 2023. The target of that tournament was alpha amylase, which is an industrially relevant enzyme. And again, we challenged the participants to not just improve the enzyme specific activity, but also improve uh, also maintain expression and thermostability. So these are some of the top participants of that pilot round. And you can see that several of the top participants were able to beat the wild type level of activity, while several of those sequences also maintained expression and thermostability. So this was openly published in the journal Proteins, the same journal that highlighted the results of every CASP competition. And then that led to a side-by-side comparison of very innovative and several novel kind of uh, generative as well as predictive AI models, including protein language models, GNNs, RNNs, et cetera. And then in total, we tested 1,200 participants submitted sequences experimentally, most of which were very unique sequences that were submitted. Again, so this benchmark was open and transparent, so all the teams submitted abstracts, so you can look up what approaches they used. And then it was reproducible. So Cradle Bioscience did a retrospective uh, um, benchmark. They took all of our published sequences, and they used their own biofoundry to test um, the results, and the results basically were reproducible even in their setup. So coming to the 2025 protein aging tournament, the target for the 2025 tournament is PETase or PET hydrolase. And again, this is like a real world impactful target. So we all know that the global plastic pollution problem is in a crisis mode. We produce 410 million metric tons of plastic every year, which is projected to triple by 2060. And majority of that plastic is not recycled. So only 10% is truly recycled. Most of it is deposited in landfills and pollutes our land, environment, forests, as well as water ways. So one of the green ways to recycle plastic as well as degrade plastic is enzymatic breakdown of PET into its monomers by PET hydrolase. Um, but there are major key issues that hinder that industrial large-scale use of PETases for recycling. The key among them is thermostability. The PETases need to act at above the glass transition temperature of PET, where the PET chains are more accessible. They also need to be active at high and low pH conditions. And of course, for any large scale industrial application, it needs to be able to be produced in large scale in a heterologous host like E. coli or Bacillus. Um, so we would be in this year's tournament, the tasks would be designed around improving these properties, both predicting as well as improving these industrially relevant properties. Um, so in terms of the structure of the tournament, we have it again divided into a predictive phase and a generative phase. In the predictive phase, again, we provide an optional training data set. Well, first, so that's divided into two tracks, a zero-shot track and a supervised track. In the zero-shot track, we have not yet released the training data set. In the supervised track, we release the training data set that the participants can use optionally. And then we ask them to predict PETase properties, multiple properties of PETases um, um, of a test set where we have withheld the property labels and they predict just using the sequences. Um, and again, top teams advance to the generative phase. In the generative phase, the participants submit sequences that have enhanced properties, and again, they can use the training data set for training their generative models. The generative phase we have divided into two rounds to pretty much mimic like a DBT cycle that is usually used in industries where you generate a set of sequences and then learn from those set of sequences and further improve your sequences. And so that's exactly what the participants will do. They will first submit a set of sequences which we will test and release the results of. And then they have a chance to further improve not just their own performance, uh, but then also improve their performance against other participants' submitted sequences. Um, so in terms of the timeline, the registration ends by October 17th. And then the predictive phase, zero shot phase starts on November 3rd. And then the generative phase starts at the beginning of next year. And then that will end around mid-April of next year. And then sometime in the summer, we'll be announcing the winners of the tournament. So again, in terms of training data sets, we will be providing multi-parametric data set along activity, thermostability, and expression. The first set of data set we'll be providing during or right at the start of the supervised track will be single mutant data of centered around a couple of PETases, but also a large sampling of natural homologs. So you have a very rich and diverse data set that you can use for training. And then right before generative round two, so that same data set that the participants can use also for training their generative models. And then right before the start of the generative round two, we'll also release the results. The teams get the experimental results of the round one sequences, so that can also be used as additional training data for improving their models' performance. The activity will be either measured at a low or high pH, or high or low pH, and either at room temperature or at a high temperature, which is above the glass transition temperature of PET. And then the expression is either measured in E. coli or using in vitro transcription translation system. A detailed experimental protocol will be released right after the registration closes, so that participants can exactly know what exactly were the expression conditions, what exactly was the protocol for assaying the various sequences. Just to give a bit more detail on the tasks and the evaluation, of course, in the predictive round, you predict sequences, predict properties of sequences that are provided, but specifically specific activity, thermostability in the form of either TM or residual specific activity after the samples have been heat shocked at 25, and then expression titers. So you predict those three properties with or without training data. So zero shot, we don't provide the training data. And then in the supervised track, we provide the training data. And that is evaluated using normalized discounted cumulative gain, which is basically a score where we rank the test set of sequences where we know according to the actual experimental determined properties, and then we compare the predictions to this ground truth ranking, and the participants that submit the ranking that matches the best to that ground truth ranking would perform the best. And then for the generative round, sorry. For the generative round, we basically ask the participants to improve PETase specific activity or thermostability and expression. And it needs to be improved above established benchmarks. So either it could be the best enzyme that currently exists that has already been engineered, or the improve or like existing best naturally occurring enzyme. So, and the evaluation there is a fold improvement or a benchmark, where again we average the scores along the three parameters to generate the leaderboards. Again, we would release more details exactly of the code that would be used for both of them, so the participants have clarity on exactly how they will be evaluated. So again, what is the incentive? Why participate? There are several. I mean, the main incentive is to solve the world plastic crisis, which we all, of course, want. Make this world a more sustainable, greener place. But outside of that, we of course recognize the top teams on the leaderboards. So the participants may participate anonymously, but then once they become the winners, the top teams are recognized and de-anonymized on the leaderboard. We have a total cash prize of $30,000 distributed among different sub awards throughout the tournament. So they have opportunity to win cash award. We of course uh, the participants can submit. All they have to do is submit sequences in the generative round, and we pay the cost of synthesizing the DNA and experimentally testing all of their sequences. So they can basically, if you specialize in machine learning and don't have the lab resources, we do it for you. So that is also an incentive. And again, we want the best ideas to succeed, and that's why again we provide the DNA synthesis and experimentation. But this year we have also started providing compute. So the teams are not limited by resources. They can avail free compute in the form of ESM3 GPUs, generously provided by our sponsor Evolutionary Scale. And then opportunity to publish the results. So the top teams will have the opportunity to highlight their methods in a special issue of Proteins, where we retrospectively talk about this year's tournament. And then we will also be inviting the top teams' team leads to give a talk at a AI/ML protein design focused conference next year. So yeah. Again, this is just laying out the same timeline. The registration would close in mid-October, with the predictive zero-shot phase starting at the beginning of November, followed by the predictive supervised track. The zero-shot is optional, so it's not required to. The supervised track is what we'll be using to evaluate the top teams, which will advance to the generative phase. But we will be incentivizing cash awards for the zero shot. So that's an incentive to participate. And then the generative phase round one will start at the beginning of next year, and then round one will last until end of January. And then round two will begin sometime in March. It'll go on until mid-August. There's gap because, of course, we have to get the DNA synthesized and test the sequences. So it takes time. Yeah, in sometime in summer, we'll be announcing the winners. But we have a very all of this is made possible by our tournament committee, as well as our sponsors. Our tournament committee consists of academics as well as industry folks who are either in machine learning, AI, ML space, or in protein engineering space. And they help us guide and determine some of the key evaluation metrics and all of the meat, the nitty-gritties of the tournament. So we're very kind of a shout out to them, very grateful for their time and efforts. And of course, this year, our Evolutionary Scale is our sponsor for providing free compute to the participants. And then Twist Bioscience is sponsoring free DNA synthesis. So all of the training data sequences of both natural homologs as well as mutants, as well as all of the participant submitted sequences. All of those DNA sequences will be synthesized by Twist. So again, very, very grateful for them. Yeah, I mean, I'm open to any questions you might have. I would highly encourage uh, your labs teams to register if they're interested in this. It's a very exciting opportunity. So thank you all for listening, and we'd love to hear questions.
Thanks, Bijoy. So now we do have time for questions, and I think we'll start by one that is in the chat from Elizabeth Stewart. Beth, why don't you go ahead and ask the question?
I'm curious about what Align does to help or split or whatever any intellectual property that comes from these discoveries.
Yeah, I think that's a great question. So, Align does not, so any new IP, like material IP, like be from sequences or any method, a process IP, if you have a novel AI/ML method, that belongs to the participants. And they're responsible for pursuing it from the time of when we start the tournament to the time where we'll openly release the data. There's quite a bit of time, so the participants can, if they think that this is worth pursuing IP, they can file patent, and we are the data that we generate. We do own that data, so we are happy to provide a limited use license if the participants want to pursue or use that data in their the provisional patent claims. So, but we don't claim any IP on participant generated sequences. That's their work, and they should be able to pursue IP on that.
Do you give, in the spirit of, education, do you give any sort of guidance in that? I could see that some teams might be very, educated on what to do, and some teams might have absolutely no idea what to do with any discoveries.
Yeah, unfortunately, we are a non-for-profit. I mean, our main objective here is benchmarking, and then seeing how the models perform, and having it openly available to the public. Don't So we are not well resourced to provide that information or help in that regard. I mean, of course, like I said, we can provide limited use license for the data if that helps in having the patent make a case and be stronger. So.
Okay, thank you.
So there was another question kind of following on to that question about IP. Do you want to jump in here, B. Mullins?
Hey there, everybody, and thank you, Joy, for coming and talking to us. Hopefully, y'all can hear me. I'm at my work right now. I just wanted to join because I was interested as soon as I saw the email. But my question was similar to Ms. Stewart over there. Is that the email said that it says why compete and retain your IP and patent your protein. So you're saying that y'all have unlimited access to the data that's derived from this competition for your own free use. Did I get that correct?
So yeah, after that is correct. So after we publish the results, so after the tournament is complete and we openly publish the results, we also make the data available on the data portal, and that is open source data. So yeah, people can use that data, however they want.
Understood. Thank you.
So some more questions. Thomas.
So, I'm going to start with the confession that I signed up right after the hackathon for this, but I lied and I said I have a team of 3 to 5 people because it was only me. And I was going to use this for a teaching opportunity once my semester, which now has started, and to recruit my research students and some that were doing work with me to use it as a teaching moment. So I can go back and just, so I'll be the team lead, obviously. So in this, I'm looking forward to using this as a way to explore my pedagogy and authentic opportunities to do work with students. So my questions come from an academic perspective. So like which we probably won't be, I don't even know if we're going to be able to do with the whole competition, but if say miraculously we actually, went on to the second round, we won an award. One of the things is and when you're at academic institutions, you're working with students, how do you split prize money? How do you make sure that you're not are there any mechanisms that, like how do you use that money, whether, to split with students or so on, or does it go back to the college? Like that'd be one of my questions. And two is, if it's okay to use this as a teaching moment, and even if you don't successfully make it through the stages, can it's still okay to try your best to do something with this program?
Yeah, so I mean that's a great question on both fronts. I'll try my best to answer it. Currently, we have, and this might change. We don't have any restriction on how the prize money can be used. And how it would be divided would be mostly up to the teams to decide. So it can be used like for internal, like the schools, the school itself might have some restrictions on that, and so you might want to refer to your organization. And that would be my thoughts on the prize money. In terms of using it as a teaching opportunity, yes, absolutely. You could use this. I mean, all you need here really is a computer and some knowledge of AI, ML, or just any protein modeling approaches, and you can compete. I mean, of course, a lot of the tasks are challenging and require time. So that's the only sort of hurdle. But we try to provide training data for recognizing that not everyone has access to high quality data for training their models. And we also are providing compute so they can avail that. But unfortunately, at this point in time, we are outside of that. We're not providing any kind of other resources that educate, so to speak. So might change in the future where there is additional sort of publications and resources that they can learn about protein engineering. So to an extent, these would be self-starter teams that are motivated towards solving these problems and are self-taught or have some background in it.
I do think, and I'll put the link here in the chat, if you're setting up a team, definitely look at this AI.gov Presidential Challenge Initiative, because that might be a way to get some funding to pay for some of the time for your students and participants. I think that would be helpful. Some more questions. I imagine a lot of people are just going, what? What do we do? Where do we get started?
I mean, you can scan this QR code and sign up.
That's right.
Sorry, I just had one more question.
Yeah,
So this
go
is
ahead.
more of a data gathering expedition, if anything, than the tournament, because there's no, like, once, let's say that we do go through the process of engineering the protein, and then it's on the open source, there wouldn't be any stopping anybody from going in, even while it was in the process of getting patented, from people using that data, correct?
I mean, so before we make it public, there is, the generative phase starts at the beginning of the year, and there are 6 months before which at least 6 months before which we'll make it publicly available, the data, and that is enough time for if the participants see a material or process IP value that they can file provisional patents for those sequences. Um, so, so yeah, I mean but after they're, I mean the public open source. I mean, so just to clarify a little bit, it's not that the data is just out there that can be downloaded publicly. You still have to create. So we still have to provide you access to the open data, and we don't. We have a different review process for-profit industry people who are asking access versus like academic teams or academic people who are asking access for research and development purposes versus like for-profit purposes. Yeah, we do own the data, but and it is openly available, but not necessarily just for profit purposes. But again, like there is opportunity for the participants to file IP, just like I elaborated earlier. So.
Gotcha. Thank you so much.
Okay, there's a question from Laura Govia, Laura, why don't you jump in?
Okay, so I'm Laura Gobea, and I teach the biomedical program. I teach from freshmen to seniors, and my seniors and perhaps some of my juniors, they are very well skilled in the lab analysis, and I would like to know if they can participate in some way doing this project. They are very, very enthusiastic about the plastic, the microplastics. So I would like to follow with you and perhaps with Dr. Porter about how if there is any way that my students can participate.
Yeah, in terms of all of the experimental work, we are using our experimental partners, either CROs or contract research organizations or other biofoundries, similar partners that perform the experimental work. Again, we want to make sure that the data is reproducible and there is integrity in the data that we put out. So as far as helping in doing the experimental work, probably not a lot of room here, but of course, if they're interested in plastic degradation and have ideas about protein engineering, they can of course participate in the tournament and try to win the challenges based on the tasks that have been laid out.
Dr. Govia, I think maybe we could help you find a community college partner. I'll talk more with Linnea Fletcher about that.
Yes.
But
okay.
I think there's some possibilities.
Okay. Thank you.
I think that would be great.
Yeah.
Are there more other questions? We have time for a couple more. Okay, I have one. I was wondering, so you're going to be working on the PETase project this year. Do you have ideas about what you might be doing next year?
Yes, we have we have actually a lot of ideas. So while we did some of the due diligence for this year, determine this year's target, we also had a very, very close seconds and thirds. I cannot like tell you at this point because like it's a matter of internal deliberation. But we are always open ears if you want to nominate a target, if you think something is I mean, of course, you would say antibodies, which is very exciting target, of
How'd you
course.
know?
Um, but and you know, very valuable from the biomedical standpoint. So, but you know, happy to. In fact, I did solicit a lot of external input in term before determining the target. So the only other thing we consider is whether we can generate high quality data on that target, and there are existing methods that can be used for validation. So,
Oh, great. Beth.
So the current administration seems to be focusing a little bit on biotech in terms of country security. And I was wondering if you have any thoughts about possible impacts on this in terms of that, because, you know, in any kind of good sci-fi novel, if a plastic eating bacteria was to come loose, you know, the world would come to a crashing halt. Do you see any issues with that?
so most of our procurement is focused on sort of industrial applications. So like not bacterial. So a purified enzyme that is like just the protein. And of course, like a lot of these, you know, plastic is hard to degrade. Like it's not something that, you know, a lot of these industrial processes are under stringent solvent, high pH conditions, and that's what the tasks are designed around. It's not something that we think that like a bacteria or even just the purified material can be used to like damage something or for nefarious purposes. That being said, we, as a part of our standard process, we screen sequences for like say someone had to submit like some biosecurity threat kind of a sequence. We screen those sequences just in a standard manner to make sure that there is no um, sequences that are of that can harm anything.
Thank you. These are sort of a bit tangential to the actual science per se, and yet they are becoming, you know, just so important and possibly impactful. So I was just wondering how you dealt with that, and it's an education moment too, to be able to say that that's what you do to the people who are going to participate. So thank you.
Yeah, I mean, biosecurity is always top on our mind, and of course we incorporate that very relevant in our processes. Yeah.
All right. So, other questions? Well, I have a feeling people may come up with some later, and I know you gave us your email. I will put that on the website when I post the recording of the talk.
Awesome. Thanks
like
a lot.
yeah.
Thanks for listening. Thanks for all of these amazing, insightful questions. Like, it also becomes a food for thought for me. So, thank you for having me.
Yeah, I think everybody appreciated it. We are just to remind everybody, we are Innovate Bio, the National Advanced Technological Education Center for Biotechnology, funded by the National Science Foundation. I have to thank our funder. I guess I have one more question. Is there anything you point people to for like getting started?
In terms of registration or
No,
in
no,
terms
in
of
terms of they want to design sequences what would you say? Where do they begin? I know they're going to have to teach themselves a lot, but
Yeah, I think that's a great question. So a lot of, for example, David Baker's work, a lot of that kind of like RF diffusion, all of those sort of models are out there. Like, so I would say just do some research and you can, uh, a lot of the things are out in the public, so you can sort of search protein engineering models, generative AI models for protein design, and get started there. But that's a great point. So maybe we could think about a sort of a starter pack, like our
Mhm.
starter. I wonder whether you have something like that with the antibody hackathons.
A little bit.
Mhm.
We have our GitHub instances. There were 3 teams working on AI this past summer, and
Mhm.
I think Thomas's is one of them. And we may have some materials that could be useful here.
Yeah, I mean, it would be
Sandra,
just to understand what are the few things that you provide would become like a good starter pack. We do plan on incorporating baseline models,
Mhm.
so that gives the participants like something to sort of uh, judge their uh, approach against. And but these are like just simple models. They're not like the best that's out there, and that the participants can just understand what that is and what that entails.
could I quickly just ask a clarification, like the couple of minutes that we have, slash question, piggybacking on Sandra's question?
Yeah, go ahead.
Yeah. So I just want to make sure I'm clearly understanding this because I don't want to. Sorry for being a little spastic. So the overall goal is to get the sequences, the new protein sequences for this enzyme to better degrade plastic. You provide some machine learning, like some tools for us to use the learning AI to generate these sequences for you to then experimentally test. So my approach to this was not just to do that, but I wanted to also, since I'm very new to all of this, try to see then how I can look at using what you provide us as a base to do that, but then also try to expand and see how students that I recruit and myself, who is very, very not needs to, do the AI, machine learning, coding, music for dummies, then to eventually look at how we could use sound and AI to generate sequences and understand more about protein structure. So it's not that you have to develop. I mean, you're using these methods to look at sequences, but you could also look at ways in this competition when you want to get those sequences to also look at ways you can improve on the methods to get to that end part to the end product.
Yeah, so um, yeah, there's a lot to unpack in that question. We provide is the training data that the participants can optionally use. They don't have to use it, but we provide as far as comprehensive as possible training data set that they can use, because we understand that not everyone has access to good quality data. So that's one part. And then the participants are free to generate a modeling strategy that can predict, or in the second phase, generate sequences, improved sequences. And then we either in the predictive phase test the predictive power of their models, or in the generative phase test the power of their models to generate enhanced sequences with improved properties. And then we test those sequences in the generative phase experimentally. So in currently, we don't have the capability of like sort of like in automated fashion test their models where they input an algorithm and then basically evaluate the algorithm. All they had to do in the predictive phase is submit predictions, and in the generative phase submit sequences that we then synthesize the DNA and experimentally test. In future competitions, we might incorporate where they had to submit their actual models, and then we run those models side by side with other teams' models. But at this point, that is not how we have designed the challenge to be.
But it can work as a nice resource for us that if we don't know whether or not we're going to get something that's, good, that a good prediction, we could use as a proof of our concept that we think this might, and then you're like a second backup saying, oh, this actually was a good prediction based on what we know, and then we could use it as a tool to help us grow as well.
Oh, absolutely. I mean, the whole idea of this benchmark is to see how different innovative approaches in predictive modeling or generative modeling, how do they stack up against each other? And so at the end of it, what we really hope is the community learns and understands what approaches are good, what approaches are not good, how do they stack up against existing methods, and then that's the real hope that and then using that collective information, the field can advance.
Thank you. It gives me hope.
Thanks. Thank you. Thanks for the questions.
Okay.